Thanks also to Prodotti Gianni for organizing this event. So as Katerina mentioned, I'm going to talk about 3D genomics today and also the solutions that Arima provides you to study 3D genome structure. So I hope that during this webinar, I will provide you with enough insights so that you can learn how to apply 3D genomics and the Arima solutions to your research projects. This is the outline of my presentation. So I have divided in 3 parts. The first part, I will talk about what is 3D genomic and also how the Arima HiC technology works. Then I will provide you with some examples on how to use 3D genomics just to study disease mechanism, also to identify potential therapeutic targets and response, and also how to use 3D genomics to detect clinically relevant genomic rearrangements. The last part of my presentation will be focused on giving you some information about the product and service portfolio that Arima provides to study 3D genome structure. Who is Arima Genomics? So Arima Genomics is a leader company in the 3D genome space. And then basically our goal is to provide solutions to accelerate the understanding of the 3D genome structure and to drive discovery and also help researchers to improve and doctors to improve human health. We're a San Diego-based startup company with about 45 employees right now. We have 6 products on the market, and we have about 600 different labs using our technologies and more than 300 publications citing the use of our technology. We are a very small team in Europe, mostly commercial team. And then we have also a network of distributors like Prodotti Gianni, who is our exclusive distributor in Italy. So the first question you will ask yourself is, okay, what is 3D genomics? What do you mean with 3D genomics, and how can I use that in my research projects? So it is well known that the genome structure and 3D genome conformation has a big impact on cellular function and specifically in gene regulation, DNA replication, or DNA repair. You probably are familiarized with a lot of different NGS-based methods like RNA sequencing, DNA methylation, or ChIP-seq. But all these methods have something in common, that they look really at the DNA, or they interrogate the DNA in a linear way, meaning that they look at the sequence and they see how the sequence is involved in different processes like disease. However, we all know that the DNA is not organized in the nucleus in the linear way, but it's really organized and compacted, and it's formed different 3D genome structures. So what 3D genomic involves is the study of not only the DNA sequence, but also the DNA structure. Looking more in detail here, we know that the higher level of organization of the DNA is the chromosome, and as you know, the chromosomes are in the nucleus, and they are forming, or they are located in different chromosomal territories. Within the chromosomes, you can find different 3D genome structures like AB compartments, which basically refer to active and inactive chromatin, or open or closed chromatin. But also you can find other 3D genome structures like topological associated domains, which are self-interacting domains, and also chromatin loops that are responsible, for example, of promoter-enhancer interactions. It is also known that all these 3D genome structures harbor biological information, and they are also involved in different pathological processes. So what do we offer at Arima Genomics? So we offer a very unique HiC platform, HiC assay, which is a high-throughput chromatin conformation capture, which allows you to study how spatial and temporal changes in chromatin conformation alter gene regulation and cellular function. So what is a HiC? Basically, this is a combination of a proximity ligation assay and NGS approach. And what you really allows to measure, what HiC allows you to measure, is the frequency in which 2 DNA fragments are associated. So basically, what you are generating when you perform a Hi-C is the interactome in your sample of interest. You know, when using other solutions, omics solutions, that, for example, with RNA, you generate a transcriptome. With DNA methylation, you generate a methylome. And as I mentioned, with Hi-C, you are generating an interactome. So how does it work? The workflow is very similar to other conventional NGS approaches or omics, but what it differs is the sample preparation. Because Hi-C measures basically frequency in which two fragments are associated, and it really reveals information about the chromatin conformation. You are really start the workflow work with at the chromatin level. So the first step of this workflow or protocol is just to fix your sample of interest with formaldehyde, the same way that you will fix your sample when performing chromatin immunoprecipitation assays. Okay. So you can work with different type of samples, and the Arima Hi-C workflow is compatible with cell lines, primary cell lines. You can also start working with blood samples. You can also use tissue samples, even FFPE samples. So the first step, as I mentioned, formaldehyde and cross-linking fixation, so that you can capture all the interactions that are happening at the chromosomal level. The second step is to perform the Hi-C reaction. And for that, Arima Genomics is offering you a very unique and patented cocktail of restriction enzymes that will allow you to digest your fixed chromatin. After that, you will fill the ends and then mark them with biotin, and then you will ligate those fragments that you have generated with a restriction enzyme cocktail. So basically, what you will ligate are fragments that are close in the 3D genome space. So the ones that are in the high proximity will ligate with a higher frequency. And basically, you are generating chimeric fragments that are representing two fragments that are close in the 3D nucleus space, meaning, and that's how you are generating your interactome. Once you have your chimeric chromatin fragments, you will purify them, and then you will share them with a Covaris or BioRaptor device, and then those fragments that has been shared and biotinylated, you will pull down with streptavidin bits. Basically, the biotinylated fragments are the ones that contain the 3D genome structure information. Then you will perform a standard library prep protocol, and then you will sequence your chimeric fragments with the Illumina technology. After that, you will align those chimeric fragments to your genome of reference, and that's how you will identify which fragments and the position of the fragments that are interacting in the 3D genome space. So why is the Arima Hi-C technology successful today? So basically because we presented the technology and a workflow that has overcome some of the challenges that are of using conventional methods or homebrew Hi-C methods. Okay, the first one is the lack of versatility. Traditionally, when you are performing homebrew Hi-C experiments, you really need a lot of cells, millions of cells, just to perform one single experiment. With Arima Hi-C technology, you can just use as little as 50,000 cells just for your genome-wide Hi-C experiments, which open the door just to use this technology with clinical samples. Another challenge that is overcome by using the Arima Hi-C technology is the time. You know, traditional and conventional Hi-C homebrew methods need about 3-4 days just to complete the protocol, while the Arima workflow takes only 1 or 2 days just from a sample prep to sequencing. The third aspect that is also improved by using the Arima Hi-C technology is the data quality. We have done a lot of experiments comparing our technology with the homebrew Hi-C experiments, and then we observed that there is a higher percentage of long resistance interactions with the Arima Hi-C. This also brings you to this is due to the chemistry, patented chemistry, and our multi-restriction enzyme cocktail that we provide you in the kit, and it also allows you to detect same chromatin loops with less sequencing depth. This brings me to the next challenge that is overcome by using the Arima Hi-C kit, which is the cost of the sequencing. You know, traditionally, Hi-C, one of the hurdles of performing Hi-C experiments is that it requires billion of reads just for per sample. But with the Arima technology, you just need 300 to 600 million parent reads per sample, depending on the resolution you want to achieve. You will tell me, okay, that's still a lot of sequencing to do. And if you compare to other omic technologies, it is true, but it really reduces substantially the need of the sequencing need if you compare our technology with traditional homebrew Hi-C methods. The last but not least, troubleshooting has been always been very hard when you perform Hi-C traditional Hi-C experiments. But with the Arima Hi-C technology, this challenge is overcome because we provide you with a very simplified Hi-C workflow, just only 6 hours of workflow, 8 steps, all in one tube. And then we have also design building QC steps within the protocol, so you can track the quality of your samples along the workflow. What is our offer? Basically, we can supply you our technology as kits, but you can also send that samples to our facilities as a service project. Okay, both options are open to you. And then here in Italy, we have a Prodotti Gianni just to supply you with our products. Just before I give you some examples on how valuable could be the Hi-C data for your research, I would like to stop here and talk a little bit about how the Hi-C data are visualized, because this is the type of graphics and plots that you will find in literature when you look or you do your research on what is Hi-C. Okay. So there are many ways to visualize the Hi-C data, but one of the most common is to use what is called Hi-C heat maps. Here, I'm showing you a heat map that has been used by the Telomere to Telomere Consortium to scaffold a complete human genome sequence. What you are seeing here is each of the chromosomes line up across the top and side. And each of the red points represent these chimeric reads that are informative of an interaction. As you can see in this graphic on the left, mostly of the reads or the red dots you observe on the diagonal, meaning that mostly of the interactions are taking place within the same chromosome. This is normal in healthy samples because, as I mentioned at the beginning of my presentation, chromosomes are located in the nucleus in chromosomal territories, and in generally, they do not interplay within each other, okay? Only when there is in disease cases like is the case of a cancer. Now let's have a look to differences between the normal karyotype and then an abnormal karyotype from a cancer cell line. As you observe in the normal karyotype, similar to in the previous slides, all the interactions and all the reads are you can find on the diagonal, red diagonal that is shown in this Hi-C heat map, meaning that mostly of the interactions are intrachromosomal. That's normal in a healthy situation. However, in a cancer, some of the events that are very common in cancer is rearrangements in the genome, and then you can use these Hi-C heat maps just to detect those rearrangements, as it is shown here in the abnormal karyotype graphic. As you see, there are some red spots, and then basically interactions that are shown of the red diagonal, which really are representative in this case of translocations happening between different or interaction between different chromosomes. This is just an example of translocations, but obviously with a Hi-C heat map, you can really detect any type of rearrangement happening between different chromosomes or within same chromosome. You can also zoom in and out these Hi-C heat maps just to look at the specific regions depending on the resolution you are interested in. Here there is an example of an interaction, some interactions happening at in one region in chromosome 2. So let me just find the pointer, laser pointer. So here, if you look at this map, at this graphic, you could see here that in these interaction domains, there are happening some interactions. This is one interaction. You see here the red, the reads that are informative of the interaction that are happening between this position, yellow, and also the green position. The stronger the color is, the higher is the frequency of these interactions. At the same time, you can see that there is also small, not that strong, but also you can see some interactions between these other domains, which are here shown as green and blue in that area, and also a very strong interaction again. You see here the red color between these other domains, blue and black, sorry. So basically, the model that you will see here in this Hi-C heat map is that there are some areas in chromosome 2 that are interacting and are generating these loops of these chromatin loops, which is these 3D genome structures that I was explaining in my previous slides. This is all about this first part of the presentation. Now I would like to give you a few examples on how 3D genomics can be used. First, to define or disease or study disease mechanisms. Also, to study therapeutic targets. And last but not least, also to identify genomic rearrangements. But before that, I would like also to stop in this slide and provide you some information about the main applications that the researchers are using when they have a Hi-C data. Okay, the nature of these Hi-C chimeric reads allows you to do the following. There are many researchers that are using Hi-C to do genome assembly and scaffolding projects. This is mainly plant genomics, animal genomics, and some consortia that are performing de novo sequencing by combining long read sequencing backed by Oxford Nanopore together with a Hi-C technology. So I would like to mention here that the Arima Hi-C technology has been used by some of the most important genome assembly consortium that are happening today, and is the technology of choice by the Sanger Institute for the genome assembly projects. The second application that is very interested when doing Hi-C is to understand genome organization. I have spoken a little bit about the use of Hi-C to detect the AP compartments, stats, and loops during the first part of this presentation. You can also use Hi-C to interpret disease risk variants, basically link non-coding disease associated risk variant to target genes. You can also use the Hi-C to discover the mechanism of gene regulation. There are a lot of labs that are doing now multi-omic approaches by combining DNA methylation, RNA sequencing, and also Hi-C to understand gene regulation. And last, and also very important, you can use Hi-C to detect structural variants in and specifically gene fusions and translocations. It's a very sensitive technology, and it's applicable for researchers that are studying cancer research or rare diseases. I just want to mention here that Arima Genomics has spent during the last 2 years a lot of efforts and research to use Hi-C for detecting structural variants, and then we have been involved in a lot of different collaboration with hospitals in US. Let's start first with the first example on how to use 3D genomics to understand disease mechanisms. This is a really exciting paper which was published in Nature Genetics by Clockton and colleagues. And here, what the researchers are demonstrating is in this paper is they are looking at how the 3D organization of an oncogene relative to its enhancer could affect the progression of T-cell acute lymphoblastic leukemia. So if you look in the upper diagram, we show you there what is the basically the conformation, the 3D conformation of the oncogene and enhancer, a distal enhancer, which is around 2 megabases far from the oncogene. And as we can see in the healthy patient, those oncogene and enhancer are part of 2 independent chromatin loops. These chromatin loops, as I mentioned, can be identified by doing Hi-C. What this researcher did in this paper was just to also analyze some patient samples, and what they observed was that in those samples, when performing Hi-C, these 2 chromatin loop conformation actually was not existing anymore, and both oncogene and the enhancer region in the presence of a specific transcription factor, they were all they were all included in 1 chromatin loop, which was facilitating the activation of this oncogene by this enhancer sequence. In that case, the oncogene is a MYC, which is also related to tumor progression. And moreover, after that, the researchers, they apply pharmaceuticals just to avoid the binding of the transcription factor to the enhancer sequence. And what they observed by performing again Hi-C is that those patients that has been treated with a drug, they were they observed that they had again a 2 chromatin loop conformation that was typical from a healthy patient. So basically, they were able to restore the 2 chromatin loop conformation characteristic of healthy patients. Being said that, the conclusions of this paper is that 3D DNA folding informs how MYC is regulated in healthy state, but also how it's misregulated in leukemia cells. In addition to that, they use 3D genomics also to understand which transcription factor was involved in the activation of this enhancer of this oncogene, and then which one was target for treatment. And last but not least, 3D DNA folding analysis also informs how MYC regulation and expression is restored after treatment. Let's jump in a second example on how to use also how valuable is 3D genomics, in this case, just to identify potential therapeutic targets and response. This is also a study that has been carried by the Garvan Institute, by the group of Joanna Achinger Kawega. And basically, what this group wanted to do is just to understand the gene expression and regulatory landscape following a tumor treatment in breast cancer in breast cancer. So to do this, they just grafted patient cancer cells into a mouse model, which they treated with epigenetic therapy, a drug called the cytarabine, which is a DNA demethylating agent. And after that, they apply a battery of omics technologies like DNA methylation assay, RNA sequencing, but also 3D genomics, and they perform genome-wide Hi-C and promoter capture Hi-C, just to understand what was the regulatory landscape before and after treatment. What I'm showing you here is the results of the genome-wide Hi-C experiments that they performed with before and after the treatment with the decitabine. And as I mentioned before, the decitabine is an agent that will cause demethylation in the DNA. And what they observe, as it is shown in this graphic, is that there is not only loss of the loss of the DNA methylation is leading to also decompactation of the chromatin. Moreover, they decide to look specifically into some specific regions or genes that they were also related to good prognosis in cancer. And what they observe is, like it is shown here in the KRT8, that some of these genes that were also related to good prognosis were gaining interactions between in the promoter regions with some active enhancers, meaning that they were in the presence of this drug and after the methylation, some of these genes were gaining in enhancer promoter interactions, which will lead to an overexpression of these genes. This is the case of a KRT8, which is a member of the keratin gene family, whose misregulation is known to promote metastasis. So when the gene is overexpressed, this is really related to a good prognosis. They observe the same gain of interactions when they analyze some estrogen receptor target genes, which are also known to be related to good prognosis. So the takeaways of this study was that 3D genomics, and in this case, 3D epigenome remodeling, is an essential mechanism of gene deregulation in breast cancer. Also, that the 3D epigenome remodeling helps to understand the mechanism by which the decitabine-induced DNA hypomethylation can suppress tumor growth. And also 3D epigenome remodeling helps to explore the potential of using epigenetic therapy to target estrogen plus resistant breast cancer via DNA methylation-dependent rewiring of 3D genome interactions. The last example I would like to show you today is how to use 3D genomics to detect clinically relevant genomic rearrangements. This is a project in which Arima has been collaborating with a lot of different hospitals in US, but this case I'm going to show you today specifically refers to a patient, a pediatric patient that came to the doctor with a stage 2 glioma, and that was initially treated with a subtotal resection of the tumor. After 6 months, the patient experienced a very rapid progression of the tumor, and then what the NYU doctors, in this case Mattia Snuder, which is our collaborator, did was just to run 2 existing tests in the market. One, the NYU Genome Pack, just to analyze to identify tumor driver mutations. And the second is a panel, NYU Fusion Sequer RNA sequencing panel, that allows you to look for gene fusions. His results showed that there was no driver for this patient. So he decided, he got in touch with Arima Genomics. He knew about us, and then what he did was just to send us the sample of the patient, and then we performed Hi-C in-house at our facilities, and we were able to identify a novel genomic rearrangement involving a gene called PDL1, which is basically a gene that, when it's overexpressed, it prevents the immune cells to distinguish between tumor cells and healthy cells. So then what the Matthias Noodle did was just to confirm that this could be a candidate to target for this candidate gene, sorry, to be target for this patient. And then he performed histology, and he observed that in this patient, the PD-L1 gene was overexpressed. So what was good about this case is that it was already existing treatment, FDA-approved treatment called Keytruda, which was suppressing the expression of this gene that, as I mentioned, was involved in preventing the immune system to attack the tumor cells. And then the value of this story is that Arima High-C solution helped this doctor to rescue this patient from chemotherapy and put this patient into a existing targeted treatment. Since then, Arima Genomics has been involved in analyzing, I would say now, about 300 driver negative patients. Here I am only showing some results of the first 122 samples that we analyzed. And then those samples that were driver negative samples, we were able to identify in about 50% of the cases a novel genomic rearrangements involving some genes that are clinically actionable, meaning that there is an existing FDA-approved drug to modulate the expression of these genes. So these collaborations we are having them right now with a lot of different hospitals across the US, and it shows a little bit the power of High-C to detect novel rearrangements in the genome. Just as a summary and some conclusions about this project, High-C provides information about whole chromosomes involving rearrangements, not just the breakpoints themselves. It is also a very sensitive technique to detect large genome rearrangements. It is also a very reliable technology, more reliable than targeted RNA or DNA sequence due to the breakpoint neutral detection. And it does not really require to know where is the actual breakpoint just to identify these structural variants. It's also more sensitive than whole genome sequencing due to the proximity link reads and less sequencing that is needed to identify those structural variants. And what is also nice about our High-C technique is it does not require the investment of a new platform or equipment like PacBio or BioNano just to perform those type of experiments. So with this slide, I'm finishing the second part of my presentation, and I would like now to give you an overview of the products and services that are provided by Arima Genomics and are supplied by Prodotti, Gianni. We have 6 products right now on the market. The first one is the Arima High-C kit. This is based on our 2 restriction enzyme chemistry. This is for gene regulation studies. Then we have a Arima FFP High-C kit for structural variant detection, and it works on FFP samples. Then we have what we call Arima High Coverage High-C kit, which is based on the use of our 4 restriction enzyme chemistry, and this is the kit that is used basically for genome assembly and scaffolding projects by some genome assembly consortia. Then we have a high chip kit. I will explain you later on what is this technique about, and also some other targeted capture High-C kits like a pre-designed promoter capture High-C kit just to generate promoter enhancer interactomes, and also we can generate or produce any custom capture High-C kit for if you are interested in analyzing specific regions of the genome. Let's start with the Arima High-C Plus kit for genome-wide High-C analysis. This is a kit that works in different type of samples, tissue, blood, cell lines, whole insects. Then you can see here in this table the different specifications in terms of sample input. Also, the reads that are needed for depending on if you want to detect, depending on the type of 3D genome structure that you would like to detect, and then is also Illumina compatible. And then here you see on the right side the different formats. This Arima Hi-C Plus kit for genome-wide analysis is the one that we recommended for understanding gene regulation and epigenetic mechanism. Also discover how a spatial relationship in genome structure impacts cellular development and disease process. Also to identify all structural elements in the genome. Basically, this is the kit that you will use to study different 3D genome structures. The workflow, as I mentioned in one of my first slides, it's very simple. You can work with different type of samples. You will fixate your sample, then you perform Hi-C, then you prepare the libraries of your Hi-C DNA, then you sequence by Illumina. This is a very short protocol and involves the use of two modules, the Arima Hi-C Plus kit in combination with the Arima library prep kit. The next kit that we are offering to the research community is the Arima FFP Hi-C kit. This is also to perform genome-wide Hi-C analysis, but this is mainly focused on the use of Hi-C for structural variant detection. It works with 5 micrometers of tissue samples, and it's also compatible with the Arima library prep kit. Just wanted to mention here that for a structural variant detection, the number of reads that you will need is only 100 million per reads, and that's why, and as you know, working with FFP sample is quite difficult, and it always depends on the quality of your FFP sample. The reason why we only recommend the Arima FFP Hi-C kit for structural variant detection is because to study promoter enhancer interactions of 3D genome structures like chromatin loops, you will require 600 million per reads, and not always the quality of the FFP samples will allow you to go so deep in terms of sequencing. Again, just to summarize, it's a kit that we recommended to detect any genomic rearrangements in the genome, and it works in FFP specimens, which is even older than 10 years. Advantages of using Hi-C, FFP Hi-C kit over other technologies. It's compatible with many different type of samples, including FFP. There is no need of investment, as I mentioned before. And this is, I think, the same slide that I mentioned before. Sorry, it's a repetitive slide. And then how does this workflow work when you start with FFP samples? Because the FFP is already fixed with formalin, you don't need to do formaldehyde fixation when working with FFP samples, and basically you will just dewash your sample. You will work with you will isolate your nuclei, and you will perform Hi-C and then library prep and sequencing. This is the third kit that we recommend for genome-wide Hi-C experiments. This is more focused on genome assembly and scaffolding projects, as I mentioned, used by different genome assembly consortia. And the kit is called Arima High Coverage Hi-C Kit. The difference with the standard Arima Hi-C Plus kit is that this kit is using a cocktail of 4 restriction enzymes, whereas the standard Arima Hi-C Plus kit, which I presented in the previous slides, is based on the use of a cocktail of 2 restriction enzymes. Okay, the 4 restriction enzyme chemistry allows you to have a better coverage of the genome, and then that's why it's also more suitable for genome assembly projects. Normally, our recommendations is to sequence 200 million per gigabase of genome size of the organisms that you will be working with. As I mentioned before, this is a kit of reference used by the Darwin Tree of Life Consortium, but it has also been used by other consortia like BGP also ERGA, which is a European consortium that is also doing a lot of genome assembly projects on invertebrates and other type of organisms. And the workflow is exactly the same as the Arima standard Arima Hi-C Plus kit, which work with 2 restriction enzyme chemistry. This is just with 4 restriction enzymes, but basically the same. You will fix your sample, then you will do Hi-C library prep sequencing and then data analysis. So this is all about the genome-wide Hi-C kits. We can also offer you what we call capture Hi-C kits. And just to make an analogy, for Hi-C, it works the same way that for RNA sequencing or DNA methylation. You can perform genome-wide Hi-C analysis, but you can also design targeted approaches. And when will you need those type of approaches? So obviously, if you don't have any 3D genome conformation data, the best approach is always to start with a genome-wide Hi-C kit, just to get overall overview of what is the conformation of your chromatin in your sample. But if you already know which regions are you interested or you want to study, we can either design some capture-based approach, customize it for you, or you can also use some of the pre-designed capture Hi-C kits that we have launched, like is it the case of our promoter capture Hi-C kit to generate promoter enhanced interactomes. And one of the main advantages of the capture-based approaches is that it reduces the sequencing cost and then also increases the computational efficiency. So if you have a lot of a high number of samples, then a capture approach which will be a more affordable approach than a genome-wide Hi-C approach, which the expenses of sequencing costs will be really high. Again, 3 types of capture Hi-C kits that we can produce for you. I mentioned, and I will explain you a little bit about our promoter capture Hi-C. This is already pre-designed, so you can just find it in our website. But we can also do custom capture Hi-C kits. Either if you want to, if you are interested in studying a specific single nucleotide variants, or if you want to generate an interactome of a specific region of interest. The promoter capture Hi-C kit, this is the one that we have already pre-designed, and then you can, we supply it in different formats, 8, 16, 32 samples. It works normally with 1 million cells, but we have some data with a lower number of cells, but it will require multiplexing of samples at certain level. And then it allows you a resolution of 3 kb resolution, and the sequencing need is 100 million per enriched per sample. If you remember when I showed you this slide for the Arima Hi-C Plus kit for 3D genome structures and for chromatin loops, which really represent promoter enhancer interactions, you will need 600 million per enriched when you perform genome-wide Hi-C kits, Hi-C experiments. But with a promoter capture Hi-C kit, you can reduce the sequencing need to just 100 million per enriched per sample. So this is what you will save in sequencing cost. So when do you need to use a promoter capture Hi-C kit? When is this a benefit? When you are really interested only in characterizing regulatory differences between samples, and then you have a large number of samples. Then when you are only interested in generate conformational profiles of promoter regions, and you don't mind about the rest of the conformation in the rest in the other parts of the genome. And also it's very useful kit also to identify novel biomarkers and therapeutic targets upon drug treatments. Okay, we have 2 panels or 2 promoter capture Hi-C kits, one for human, one for mouse. And basically, this is a combination of 3 modules: the Arima Hi-C Plus kit module, then the Arima library prep module, and also the capture module, which represent the probes that you will use to analyze the promoter regions that are included in this kit. So it's about 23,000 targets that you will be able to interrogate with the promoter capture Hi-C kit, either in human and 25 with the mouse panel. also very robust and reproducible kit. We have done a lot of comparisons in different cell lines, and here is the percentage of map reads. Normally, you obtain 90% of map reads, and then also a high percentage of on-target rates. So basically, you are able to identify mostly of the reads will be informative. So this promoter capture Hi-C kit is based on our two restriction enzyme chemistry, meaning that out of these three modules, the Arima Hi-C Plus module, the library prep, and the capture module, which is the RNA baits, which is our two restriction enzyme chemistry. It allows you to get to a 3 kb beam resolution, but this always depends on the bioinformatic tools that you will use, and also allows you to multiplex multiple Hi-C libraries per capture reaction, which will also help you to reduce the cost of the experiment. The workflow here for the targeted approaches is a little bit different to the standard genome-wide Hi-C approach, but it starts the same way. You have your sample, you will fix, you will perform Hi-C. Then you prepare your library, you will amplify your library, and then you can also perform some shallow sequencing as a QC steps. And once you have your amplified Hi-C libraries, then you will use your capture module, which contains your RNA bait or probes, just to pull down those regions of interest that you want to study. So if you would like then, or if you are interested in studying a specific region that we don't have a capture Hi-C kit, or we don't sell a capture Hi-C kit, we could design a custom capture Hi-C kit based on your interest. Okay, and in that case, we have 5 different tiers, which represent 5 different pricing tiers. I would say that it will depend on the genomics, the size of the genomic region of interest or you want to study, and also the number of probes that we will need to design just to capture the region of interest. And it's very similar to the promoter capture Hi-C kit. Normally, it works with also 1 million cells, and the sequencing requirements here is 10 million per enriched per megabase of capture region. The last kit I would like to introduce you to is our Hi-Chip kit. This is a very unique technology which combines the use of Hi-C and chromatin immunoprecipitation. I don't know how many of you are familiar with chromatin immunoprecipitation. As you know, chip or chromatin immunoprecipitation is used to generate binding profiles of a specific protein of interest. With the Arima Hi-Chip kit, you would not only generate this binding profile of your protein of interest, but you will also able to detect all the interactions in which this protein is involving. Okay, so it's again a combination of the Hi-C and Hi-Chip, and I will show you now how the workflow works. So the first step basically is just to perform a Hi-C. Then once you generate your Hi-C chimeric chromatin fragments, you will share them with a sharing device like a Covaris or Bioraptor. Then you will add the antibody of the just to capture or immunoprecipitate the protein of interest. And in this moment, you are capturing all the you are capturing of your protein of interest with all the DNA fragments, which in this case, because you have performed Hi-C, you will pull down fragments that contain the binding site of this protein plus any other DNA fragments that are associated to this binding site. Okay, so you get 2 types of information in only one assay. So, and here, this is my last slide. I think one of the last slides of today's presentation. Basically, I want to show you a comparative of different omics. And here you can see the high chip data, which are represented with these arc plots that you can see here. This rosa are plots. These represent interactions that are happening between different regions that in that case are enhancers and also the MYC promoter region. I would like also to mention that in addition to all these products, we can also offer you services. All our labs are located in California, so mainly all the work will be done in San Diego. And in this case, you only need to send us your samples, either frozen or fixed cells, and then we will be responsible and we will do all the experiments from high-C library prep sequencing and also bioinformatic analysis. With that, I would like to emphasize that, our technology is the most robust high-C technology in the market, as it has been proved by the experiments that the Sanger Institute has done. We provide end-to-end solutions. We have a very user-friendly workflows, which brings, the high-C to all the labs. We have also an integrated library prep module in our workflow. The technology that we offer and our solutions work with multiple and different type of samples, and then we have a world-class service and support. And with that, with this slide, I conclude the presentation of today, and then I leave you there some links where you can find some documentation and technical support information, and also remind you that you can acquire any of our kits through Prodotti Gianni in the Italian territory.
Thank you very much.